Good morning, everybody. Thank you very much for coming. I hope that everybody is, I'm sure a few more people are going to be sort of trailing in here as we get going, but I'm going to get started. So my name is Stephanie. I am a fifth-year PhD student in Evolutionary Anthropology, and I am here today to introduce my colleague, Megan, who is presenting today for our colloquium series. So just before we get started here, just a reminder to everybody, if you can keep your camera off while we are recording, it'll just keep the internet connection a little bit better. And it looks like everyone's on mute. And if we can keep everyone on mute, that would be great. Until the end, at which point you're welcome to turn your camera on and unmute yourself to ask any questions once Megan is finished. So I do want to start here by sort of acknowledging where we are. I don't know about anybody else, but this week in particular, I've been watching the news and feeling very grateful for being in the Southwest and having the Southwest sun and having power still running. And in that vein, I want to take a minute to just read the land acknowledgement that has been put together by the University of New Mexico, which acknowledges that the University of New Mexico sits on the traditional homelands of the Pueblo of Sandia. The original peoples of New Mexico, the Pueblo, Navajo, and Apache people, since time immemorial have a deep connection to the land and have made significant contributions to the broader community statewide. And we honor the land itself and those who remain stewards of this land throughout the generations, and also acknowledge our committed relationship to Indigenous people. And we gratefully recognize our history. So everybody, I hope, can just take a minute to feel that appreciation and feel grateful for where we are today. Megan is here today to present a talk because she has been the recipient of the 2020 Frida Butler Award. The Frida Butler Award is a scholarship given annually to honor the memory of Mrs. Butler, whose grandson, Dr. Richard A. Barrett, is an emeritus professor at UNM in our department. And this scholarship provides support for graduate research for students once they have completed their master's. So Megan, before coming to UNM, did her undergraduate degree at the University of Michigan in anthropology. And then she took a couple of years off after completing that degree and got an array of really incredible experience, including spending quite a bit of time living in Ethiopia and Kenya, where she worked as a field assistant on a baboon project with Joan Silk. And so during that time, she got to spend a lot of time observing both Hamadryas and Anubis baboons. After that, she became the lab manager for Alex Rosati at Harvard University, who was then, I should say, at Harvard University. And she, during that time, was working on cognitive evolution projects in lemurs, apes, and monkeys. And that included going to the Duke Lemur Center, going to Cayo Santiago, where she studied rhesus macaques and helped with the experiments there, and also going to Ngamba, a chimpanzee sanctuary located in Uganda. And it's actually from her involvement in that project and her work at Ngamba that she got involved in some of the research she's going to talk about today. Since being at UNM, Megan has been first and foremost a wonderful lab mate for me. Also, she has been an outstanding student and earned herself an NSF GRFP, and most recently a dissertation field grant from the Leakey Foundation. And I'll add that she earned both of those on her first try, making it like all the more impressive. So as I mentioned, the research that she is going to talk about today is from her work on sanctuary chimpanzees in Uganda. The title of her talk is Healthy Cardiovascular Profiles Across the Lifespan in Wild-Born Chimpanzees. And I think at the end of that talk, she's also going to give us a little teaser about kind of what's coming next for her in her PhD. Okay, Megan, I'm turning it over to you. Go ahead.
Great. Thank you, Stephanie, for that really nice introduction. Let me share my screen. So hopefully you are seeing the correct version of this. Yep.
Yeah, it's good, Megan.
So great. Thanks again, everyone, for being here. I'm excited today to share my master's research, where colleagues and I found evidence for healthy cardiovascular profiles across the lifespan in wild-born chimpanzees. Cardiovascular disease is an important area of comparative research because it's a major source of mortality in humans, and it's especially prevalent among adults over the age of 65. And so it's been known as a disease of aging. So the plot on the right here shows coronary artery calcification, which is one risk factor for cardiovascular disease. And as you can see, scores go up exponentially with age in many reference populations. Because humans live for a very long time compared to other animals, and they have especially long post-reproductive periods, it's been proposed that the high rates of heart disease might stem in part from our extended lifespan. So in other words, this might be a byproduct of selection for health during reproductive years. And so for some time, it was accepted as sort of an inevitable outcome of aging. But we now have several decades worth of research to counter this idea. So a lot of this comes from subsistence societies, so groups that make a living hunting and gathering, like the Hadza of Tanzania, and forager horticulturalists like the Tsimane and 𝗏𝗏 of Amazonia. And these groups appear to show minimal risk despite living to ages exceeding 70 years old. So in this figure, Tsimane is in light blue, and you can see they maintain basically zero risk all the way to age 80. So this healthy aging in groups like the Tsimane has been attributed to high levels of physical activity and diets which are high in fiber and low in saturated fats. And overall, this suggests that cardiovascular health is likely to be influenced by lifestyle. So this is consistent with what we hear from doctors all the time, that diet and exercise can help buffer against heart disease. And because the lifestyles of these subsistence societies closely approximates what we would have likely experienced during a majority of our evolutionary history, it also suggests that cardiovascular disease is not just an inevitable byproduct of aging, but actually appeared relatively recently in our evolutionary past, along with the emergence of industrialization. So this is consistent with the mismatch hypothesis, which states that rapid changes in modern environments can interact with ancient physiologies to produce maladaptive outcomes. So if we think about environments in which we evolve to look something like this, these are Hadza population eating tubers and 𝗏𝗏 participating in long distance running. But these days, at least in industrial societies, we see a lot of people spending their time doing things that look more like this. So this is an extreme version, but we see more sedentary lifestyles with processed foods. And so if we adapted to a lifestyle with endurance activity and became dependent on this physical activity, it might put us at exercise, and especially paired with this unhealthy diet. So I just want to give a brief overview of what this pathway sort of looks like. I mentioned age and lifestyle to be important predictors of these physiological risk factors, which will be a focus of this talk. So this includes what's known as pro-atherogenic blood lipid profiles. So this includes high rates of high levels of cholesterol and triglycerides, as well as low density lipoprotein or bad cholesterol, which actually is what contributes to plaque and clogging arteries. And this is often associated with low levels of high density lipoproteins or good cholesterol. We also see both interactive and independent effects of obesity and inflammation, and these things often occur together. So this coalesces, at least in humans, most often in coronary heart disease, specifically in the form of atherosclerosis or clogging of the arteries, with things like cholesterol, and again, this is exacerbated by obesity and inflammation. So restriction of blood flow is what leads to risk of heart attack and stroke. Understanding the evolutionary process, history of this process would benefit from expanding the comparative framework to include not just data from different groups of humans, but also from closely related primates, such as chimpanzees. So in this project, we ask whether aging and lifestyle effects are shared between humans and chimpanzees. Chimpanzees are a useful model for asking this question, not only because they are one of our closest living relatives, but because of this, we have a shared evolutionary history and a shared physiology. So prior work has shown from morphology, physiology, and behavior that modern day chimpanzees are probably very similar to what our last common ancestor with chimpanzees would have looked like. And so they can serve as sort of a referential model for our early hominin ancestors. We also share with chimpanzees a long and slow life history relative to other primates who are often used as models for studying human health. So if we think that selection for longer lifespans might be associated with changes in aging biology, studying this process in other long-lived species might be more informative for telling us something about human aging. And finally, in modern chimpanzees, we see similar variation in lifestyle factors, such as diet and physical activity, which we know are important influences on human health. Previous research on cardiovascular disease in captive chimpanzees has shown that this is a leading cause of mortality in captive apes as well. But they don't appear to get atherosclerosis in the same way. So this is a human artery that is atherosclerotic. Instead, they tend to get interstitial myocardial fibrosis. So this light pink is deposition of scar tissue, which can lead to heart failure. So we might be surprised then to learn that captive chimpanzees also exhibit high levels of these unhealthy blood lipids like cholesterol. So this plot is showing that chimpanzees living in a laboratory setting have cholesterol levels that are as high or even higher than industrialized human groups. But importantly, these levels don't seem to increase with age or predict fibrosis, which makes them actually an especially good system for studying cardiovascular health, because individuals with these elevated profiles aren't likely to die early like they would in humans, for example. And authors of this paper and others have suggested that this means we've inherited a high risk of cardiovascular disease from our last common ancestor with chimpanzees. But this relies on the assumption that these animals are representative of our last common ancestor. And as I mentioned, most of this research has been done in captive settings, specifically in research laboratories, where chimpanzees experience limited access to ranging and diet, which consists mainly of processed primate chow, which is shown here. Chow is low in fat, but it contains highly processed grains, which would be absent from a wild diet. We also know that captive chimpanzees tend to be overweight and obese compared to their wild or might contribute to elevated lipid profiles. And so this really means that our current reference value is representative of environments in which these physiological processes that we're interested in actually evolved. So ideally, we would want to measure this in wild chimpanzees, which would be most similar to our last common ancestor. And their diets are higher in fiber, and they have more physical activity by ranging between fruiting trees. But blood collection isn't possible in the setting due to ethical and safety reasons. Wild-born, free-ranging chimpanzees living in African sanctuaries are a good alternative for this, as their lifestyle more closely approximates wild chimpanzees, the relevant model, and blood collection is possible during veterinary examinations, which are a routine part of the sanctuary management. So we already know that the same risk factors appear not to lead to the same health outcomes. So laboratory chimpanzees have high blood lipids, but don't get atherosclerosis or these clogged arteries. But because of differences in lifestyle factors, we test the prediction that sanctuary chimpanzees have healthier profiles, especially during aging. So I'll give you a brief overview now of the methods that we used before getting into the specifics of two studies. This work was done in collaboration with Alex Rosati at the University of Michigan when I worked for her as her lab manager, as well as Melissa Emery Thompson, who is my PhD advisor here at UNM. And the first paper out of this line of research was recently published in Philosophical Transactions. So we collected biomarkers of cardiovascular health from chimpanzees living at 2 African sanctuaries over 3 sampling periods, Ngamba Island in Uganda and Chimpunga in Congo. So a vast majority of the individuals living here are wild-born. Most of them are rescues of the bushmeat and pet trade, which means that they arrived at the sanctuary at a young age, usually as infants, were quickly rehabilitated and then live the rest of their lives in naturalistic, social, and ecological environments. So they spend a lot of time free ranging in these large tracts of rainforests that you can see here. And in these forests, they eat local foods that exist naturally on the island and are very similar to what chimpanzees living in the wild would be eating. The sanctuary does supplement with local fruits and vegetables. So the photo I showed earlier was a chimpanzee eating bananas and oranges, which is sort of intermediate potentially between wild and captive, but importantly, their diets don't include any of the high calorie chow that is a staple in the laboratory diet. We studied 75 chimpanzees who were between 1 and 33 years old at the time of sampling. Chimpanzees can survive into their 50s or 60s in the wild, and sometimes even longer in captivity, but chimpanzees over 30 are considered older adults in the wild, and so this is actually a good in our sample. So in this photo, here I'm at Ngamba helping with an annual health exam. And in collaboration with sanctuary veterinarians, we collected 3 main types of data, which correspond to the major categories of risk for human cardiovascular disease. First, we ran blood lipid panels on site, measuring total cholesterol, triglycerides, and HDL and LDL, which is good and bad cholesterol. We also measured markers of obesity, including body weight and skin fold thickness. and several inflammatory markers, including C-reactive protein and interleukin-6. So the blood analysis was mostly done on site using point of care testing. The diagram at the bottom shows the process, which uses a small drop of blood, basically the amount from a finger prick, which is put into this machine, which spits out results within a few minutes. So it's a good tool to give the sanctuaries results right away. And it's good for a field setting. The first study that I want to talk about is directly compares blood lipid profiles across age and lifestyle categories by comparing the sanctuary data with a reference data set from the National Institute of Health called the Primate Aging Database, which comprises chimpanzees from 3 US who are all socially and how by the database. We didn't have individual data on things like activity level or metabolism, either for the laboratory or the sanctuary, but we have these broad lifestyle categories. And this includes 471 chimpanzees up to 60 years old. So our general approach was to ask whether there were differences with age or between laboratory versus sanctuary, and whether there was an interaction between the two. So did we see any differences increase with age? So let me orient you to these results. You'll see laboratory chimpanzees in blue and sanctuaries in green. This dotted line represents a threshold for what's considered to be healthy levels in humans. So this is a clinical threshold. These yellow bars are reference means from industrialized US and 3 subsistence societies. For both total cholesterol and triglycerides, lab chimps are higher on these levels and show a greater proportion falling over the clinical elevated threshold. And in both cases, this difference increased with age. So what you're seeing here, a different kind of plot, this is the estimate effect of age after controlling for facility. So here we split the labs up. They're now the 3 shades of blue based on the current lifestyle categories provided by the database. So one lab was indoor access with only chow diet. The second was some outdoor access with chow. And the third was chow supplemented with fruits and vegetables and outdoor access. So for triglycerides, the age difference was based on the labs increasing with age, which makes sense because this is a reflection of dietary fat. The total cholesterol, actually, we see an overall effect of decrease with age. But as you can see, this is likely to be driven by this one lab, the outdoors mixed diet. And we suspect that this is based on a change in facility management during the lifetime of the oldest cohort. So although their diet is supplemented with fruit and vegetables now, maybe when they were younger, they were eating more chow. We can also break this total cholesterol down into HDL and LDL, and remember that this is good cholesterol, so the risk threshold is actually to be below this line. And we see the same pattern of lower levels in sanctuaries. This is actually, you'll see that adults and older adults are combined here because we have a restricted sample from only one of the labs. But overall, we see a much starker difference in LDL compared to HDL. And we also see higher HDL to LDL ratios in the sanctuaries, which is cardioprotective in humans. Next, I want to talk about some new data we have on adiposity and inflammation from the sanctuary chimpanzees. So inflammation is the body's way of protecting and repairing itself. When immune cells are mobilized, they produce the redness and swelling that we're familiar with. And this process is important for fighting infection and injury. But chronic activation of the immune system can result in tissue damage, which leads to things like chronic diseases, including cardiovascular disease. And this is exacerbated by obesity. So inflammation in particular is also involved in atherosclerosis, specifically by contributing to restriction of blood vessels, but also, as you can see in this diagram, it's proposed to directly promote the production of plaque as well. Many studies from a clinical perspective has shown an accumulation of low-grade inflammation to be associated with obesity and cardiovascular disease risk during aging. So the idea here is that during the aging process, the body experiences increased immune dysregulation and loses its ability to respond in an adaptive way and relies more on chronic inflammation during aging. And this is consistent with the hypothesis that I presented earlier that chronic inflammation is just an inevitable byproduct of the aging process. We also have comparative data from subsistence societies. So, and we see sort of mixed evidence here. So some groups like the Hadza and the 𝗭𝗏 really show no evidence of chronic inflammation. On the other hand, a lot of work coming out of the Chimani group has shown a different story. So the plot on the right here is showing C-reactive protein, which is a biomarker of inflammation. And this is what they found actually is that Chimani levels are considered to be, average levels are considered to be clinically elevated. And despite this, they still maintain low body fat, low blood lipids, and the lowest reported rate of cardiovascular disease in the world. And so it's quite striking from a clinician's perspective that they still exhibit this chronic inflammation. And it's been proposed by this group that the high load of certain types of parasites, called helminths in particular, might actually be protective against cardiovascular disease in combination with healthy lipid profiles. And overall, this is consistent again with the hypothesis that inflammation levels are influenced by lifestyle factors. So this also means that high levels of inflammation during aging in the industrial world might again be due to a mismatch, this time between an evolved pro-inflammatory response and reduced pathogen exposure, which might lead to overreaction of the immune system and ultimately chronic inflammation. So we wanted to test this in chimpanzees, and returning back to this, we'll now focus on the second two, which often go hand in hand, obesity and inflammation. So this is a direct comparison again between the sanctuaries and the labs. This also includes historical data from Ngamba. And again, we see body weight being lower at the sanctuaries, and this difference increased with age. But body weight is not really a very good measure of body fatness. It's actually a better measure of body size. So we also collected skin fold thickness, which is a measure of subcutaneous fat that's used to measure adiposity in humans and other primates. And it also correlates with blood lipid levels in humans. So we took 4 skin fold measurements and created a composite skin fold thickness score. And our the first thing that we did was see how it correlated with weight. And we found a positive correlation, which suggests that it's a good proxy for obesity, and so that we could use weight in our other models appropriately. We don't have raw data from the laboratories to compare to directly, but compared to published values, which are the stars on this plot, females in sanctuaries appear to have lower skin fold thickness on this one of the measures. So this is the thigh. And this is similar for the other measures that we took as well. Okay, so let's move on to inflammation. We next analyzed levels of C-reactive protein, which is the biomarker that I showed earlier. And at very high levels in humans, CRP is also predictive of heart attack. So again, we're using clinical thresholds from humans here. So this is because these are the reference categories that we have, although we can't really draw conclusions about what is considered an elevated level in chimpanzees. But this is a good reference. So the first thing that we did was compare CRP across the 3 sampling periods at the sanctuaries, because these actually corresponded to 3 different levels of infection among the chimpanzees. So relative to a published zoo reference, we see a lot of variation, as you might expect. In the year that there was no infection, only one of the chimpanzees was sick. Heavy infection year, we saw a lot more variation. So this sort of, again, validates that this is a useful tool for identifying individual level infection at single time points. But in order to draw conclusions about chronic inflammation, we need to exclude chimpanzees who are known to be sick. So this red category. So when we remove them, and here we're looking at C-reactive protein again and IL-6, which is another pro-inflammatory biomarker that we actually measured here in the lab from serum. What you'll see over here, there are a couple of points above this clinical threshold, which all come from the high infection period. So it's possible that we're picking up actually on acute rather than chronic inflammation here. But importantly, for both of these biomarkers, we don't see any increase with age. And all of the variation, I should point out, in the healthy range is really not biologically meaningful. But the take home here is that we don't see evidence of chronic low-grade inflammation with aging. And this is actually consistent also with new data coming out from wild chimpanzees. We also found no relationship between inflammatory markers levels, and this is probably because we see very little variation. All of these measures tend to be very low in the sanctuaries. So to sum up what we found, first, unlike in humans, we didn't see consistent age-related increases in biomarkers. So in the lab sample, we saw increases in body weight, triglycerides, and low density lipoprotein, or that bad cholesterol, which suggests that this aging effect might be unique to industrial human populations. Second, we see that sanctuary chimpanzees exhibit overall healthier profiles, suggesting that this lifestyle effect is shared between chimpanzees and humans. And third, our inflammation data is consistent with some subsistence societies like the Hdl and the 𝗭𝗄, who exhibit healthy blood lipids in combination with low inflammatory biomarkers. So this is in support of the idea that you don't need high parasite load in order to maintain healthy blood lipid levels. At the beginning of this talk, I mentioned that because previously published reference data described chimpanzees as having very high lipid levels, one idea was that we inherited a risk of cardiovascular disease from our last common ancestors. But we now know that just like humans, chimpanzees don't necessarily have high lipid profiles even during aging. Our work contributes to a now very large body of research suggesting that lipid-related heart disease appears to be derived in industrialized human groups. But this still leaves us with a big outstanding question, which is why we see differential health outcomes under similar physiological risk profiles. So in other words, laboratory chimpanzees exhibit overall higher cholesterol levels, at least relative to sanctuary chimpanzees, but industrial humans are the only groups that are getting atherosclerosis. So one possibility that we've put forward is that industrial humans in particular have experienced more extreme or greater sensitivity to an evolutionary mismatch. And this might be due to recent changes in human evolution. So first, we've seen a transition towards more high quality but difficult to obtain foods such as honey and meat. And this is likely to have led humans to be more susceptible to fat storage in times of energy abundance. And so compared to lab chimps even, we see much higher levels of body fat. And we know that this can lead to obesity and inflammation, and energy abundance is really common in industrial societies. Another idea is that with the transition from more resistance to endurance physical activity, there's actually been change of the human heart. So selection for endurance activity might make us more susceptible to risk of atherosclerosis in particular under sedentary conditions. So it seems overall that we inherited from our ape ancestors a physiological system which is sensitive to lifestyle as a predictor of cardiovascular profiles, but it's the combination of these changes in heart morphology along with lifestyles that are rich in calories and more sedentary in industrialized societies that it might explain why heart disease is so deadly in these groups. So I want to spend the last few minutes talking about my future plans for my dissertation, which will be on temperament, stress physiology, and social influence in wild chimpanzees. So working on this master's project got me interested, of course, in aging, and I've started thinking more about social aging, and this is mainly spurred by another study from our group, the Kibale Chimpanzee Project, which was also led by 𝗤𝗏𝗏𝗏. The plot on the right nicely shows that older males at 𝗏𝗏𝗏𝗏, so these are wild chimpanzees now, older males are preferred social partners even after controlling for rank. So they're more likely to be in parties with other males, in close spatial proximity, and they receive high rates of grooming. So we often think about social patterns in chimpanzees being driven entirely by dominance rank. So you might expect to see young, I should say prime-aged males receiving more social attention, but they found the opposite. And understanding what makes these social partners and exploring how they might influence other members of their group really was the catalyst for the ideas that I'm going to talk about now. And this fits nicely into a field of study which focuses on the evolution of leadership across animal societies. So one big model is that social influence is rooted either in dominance or prestige. Dominance systems are characterized by active leadership based on coercion and physical intimidation, while prestige systems are characterized by active followership based on admiration. So the idea here is that you have socially attractive leaders who typically possess some important skills like foraging expertise or fishing in this example of this photo. And this makes others want to follow them. So this is importantly, prestigious leaders are more likely to be older because they've accumulated some knowledge. And current perspectives sort of suggest that dominance is a default system in animals, while prestige appears to be unique to humans. So a big question in the field is, what about our evolutionary history might have facilitated this change? And because dominance and prestige are sort of about competition and cooperation, a leading explanation is that prestige emerged along with reliance on extensive cooperation within the human lineage. So in other words, if this is more of a spectrum, which is sort of increasingly the way that researchers are thinking about this, where do chimpanzees fall on the spectrum? And is it closer to prestige compared to other animals? Chimpanzees provide an important comparative model for addressing this question as well, not just because they're one of our closest living relatives, but also because they appear to have undergone similar selection for cooperation. And so we know from prior work that chimpanzees maintain dominance hierarchies, which are based on the use of aggression, but achieving high rank is likely to be based on the use of coalitionary aggression. So this is more cooperative. They also participate both in dyadic cooperation, so close social relationships, and they also participate in higher order coordination during activities such as boundary patrols shown here in defense of community territory. So overall, this should require greater social tolerance compared to other primates. And this leads to my overarching hypothesis of my dissertation, which is that chimpanzees retain a dominance-based system of social influence, but they also exhibit some of the precursors of prestige. So in my work, I'll test for the presence of 3 key features of prestige-based influence, which are present in humans. First, humans, and especially human leaders, exhibit non-reactive temperament and increased social tolerance. So we see less behavioral reaction and hormonal reaction, for example, lower elevation of stress hormones. Second, as I mentioned, high status is more likely to be rooted in cooperation and prosociality, so things like generosity, rather than physical intimidation and competition. And finally, prestigious human leaders play a key role in enhancing group cohesion, which is proposed to be closely related to things like social learning and important for big picture questions like scaffolding large-scale cultural transmission. So this question of chimpanzee leadership style has been on for quite a while. And so we actually have some evidence, in particular for number 2, that this is probably true. So we know that some alpha males are successful through physical intimidation and more individualistic strategies, while others seem to rely more on cooperative coalitions. But my research is unique in that it asks how, well, first it breaks down this complex pathway and asks how these different strategies might be associated with underlying temperament and how they might scale up to group-wide processes of things like cohesion. So breaking down this pathway, which is really complex in human prestige, and asking whether any or which of these features is present in chimpanzees will ultimately help us to understand how the process of cooperative human leadership might have evolved. So I'll conduct this study with the Kibale from this population because they're well habituated to the presence of human observers. So we have long-term data of
Sorry to interrupt you for just a sec, but I think you cut out for the most of this slide. If you want to just repeat what you said on this slide.
This one, sure.
Yeah.
Sorry about that. So I will be working with the Kibale Chimpanzee Project to answer these questions, which has now over 30 years worth of longitude. And I want to thank now the efforts of the field assistance and data entry team in Uganda. So unlike at the sanctuary, we can collect detailed behavioral data from this population because they're habituated to the presence of human observers. And we have now really good long-term data on measures of things like social status and group cohesion, which I'll combine with collection of new fine-grained data on temperament. So we also have a very large bank of urine samples, which we can use to measure concentrations of hormones present in the urine. And I'll contribute to this by measuring a stress hormone called cortisol to understand relationships between temperament, stress physiology, and social influence in this population. So I want to thank the Butler family for giving me this opportunity to present my research. Also, so many people who've helped out with both of these projects, so funding permitting agencies in Uganda and the Congo, and many collaborators involved with research design and data collection and logistics. So I will stop sharing now, but I'm happy to share again if anyone has questions.
Yay, thanks, Megan. That was a wonderful talk. We have about 10 minutes, 12 minutes or so, which is a great amount of time for anybody who's got questions. You can just feel free to turn on your camera and unmute yourself.
Ian, I think
that was a great talk, Megan. I really love I really love it. I'm a huge fan of the study that you've published, and I'm excited to see where you go with the future work. I am curious whether or not the future work has a health angle to it, like whether or not prestige might be associated. I don't know about with markers of metabolic health, but some angle to it, some health angle to it.
Yeah, so I am continuing this line of work with the sanctuary data. So we're working on the inflammation markers, for example. The stress physiology, I think, is related to that in some ways because there are some links to health potentially, but one of the goals of the physiological data is to just get a better sense of patterns of cortisol, which we have so many samples from our group. That's a lot has been published recently, but understanding how stress physiology. So for example, are you more, do you have higher reactivity to stressors? Like, is your stress level higher during intergroup encounters or during mating competition, things like that? If you have sort of a more stable and low reactive profile, how does that link to your behavioral data? And of course, we have health measures too that we could link that to. So it's a good question. Hi, Siobhan.
Hey, great talk, Megan. That was super interesting. Like Ian, I think it's really cool that you're linking together all the kind of physiological stuff with the behavioral ecology. The human-chimp comparison is always interesting. I'm wondering, if you're thinking about, it's interesting the inversion that you're suggesting about leadership, because we often think in humans that we sort of start with a prestige bias style of leadership that then can give way to dominance or more coercive styles in different kinds of contexts involving intergroup competition and stuff like that. And I'm intrigued by this idea that leadership can arise in a prestige biased way in chimpanzees if it involves sort of coordination of group activities, right? This is something that we see in humans too, call it managerial mutualism, whatever. And what are the kinds of activities you think that are going to be sort of subject to that style of leadership in chimpanzees?
Yeah, so one thing that would be nice to look at would be during things like intergroup encounters or boundary patrols, but our group actually, it doesn't do that very often. So that might be a little hard, but the measures of cohesion that I'm looking at are include like general group levels of things like aggression and grooming. So this kind of at the mood of the group, like if they're on edge. Sorry, hopefully I didn't cut out there.
You cut out just a little bit, Megan, if you want to repeat the last little chunk you're saying about the measures you're looking at.
Yeah, sure. So we have, we can look at group level aggression and grooming to get at sort of the mood of the group. We also have party composition, so we can see like, do groups stay together for longer before they split into subgroups when certain leaders are present? Those kinds of things, as well as social network measures to see if there's like a stronger group level cohesion from like an analytical perspective.
Well, yeah, it'd be interesting if you got that kind of facultative shift in leadership style, depending on what the activities were, but thanks.
Yeah, and one other idea is that, you know, there might be simultaneously different kinds of leaders happen like at the same time in the group. So you might have an alpha who is has more of a dominant style, but maybe there's also an older male who takes on more of this prestige role, who maybe is no longer high ranking, but he's at the center of grooming social networks, and his presence has a different effect on the group. Hi, Chris.
Hi. Can I ask the next question? It kind of dovetails with Siobhan's.
Yeah, sure.
And I think, like, in terms of talking about domains and different styles of leadership, well, actually, hold on, let me back up. That was an awesome talk. You did a great job. Thank you. That was amazing. But to just ask this question quickly, we've talked a lot about males. How do females fit in?
Yes, I'm glad you asked this. And I meant to mention most research in chimpanzees, especially in the area of leadership and rank, has focused on males. So one of the goals that I didn't have time to talk about here, but is to understand how life history might play a role. So thinking about age and how maybe costs and benefits of being social at different ages might contribute to different patterns of leadership. So I mentioned like maybe younger males are taking this more dominance approach and older males might be more cooperative. But this female angle is pretty unexplored. And so another goal is to sort of probe whether females follow the same patterns. And in chimps, females seem to be less affiliative and less aggressive. So there's just sort of interact a little bit less in general. So that actually leaves a lot of room for exploration. So you might expect that they show similar patterns to males, but just in sort of lower magnitudes. But also we don't know as much about them. So that's a good area for future research.
Thanks, Megan. Just can I ask one little follow-up? Like, are there particular domains that you think like female leaders are going to be? Like when you mentioned that there are, you know, there can be multiple leaders depending on situations or things like that. Like, where do you think that females are going to like really pop out?
So in a lot of long-lived species like humans and elephants and bonobos, we see females taking a role in domains of leadership related to ecology. So for example, when food is scarce, then and whales, matriarchs are the ones who have this ecological knowledge, and so they're followed. So one of the things that I'll be looking at is followership during group movement. And so I expect that older females might be more likely to be followed, especially seasonally when there's less fruit available, or in really extreme times when the animals sort of split up. Good question.
Thanks, Megan. And again, great job. That was fabulous.
Thank you. Let me check the chat. I think I see a question here. What are your goals and dreams after your PhD is finished? What are your long-term plans? I would love to continue doing research in this area. I'm really fascinated by behavior and physiology and cognition. So I would love to look for a postdoc after this and see where that goes. Any other questions?
I have one last question, Megan, just about other comparative data that's available. You make a very good case for chimpanzees as a good comparative data set for this health physiology data, and the need to, I mean, the benefit of looking at the sanctuary chimpanzees because you have access to that data. But I was curious to know if there was other similar data sets. Like there's a lot of macaques, for example, that maybe there's lab data on macaques, and maybe 𝗂 Santiago has some health data. I don't know if they do knockdowns very often, but you know, is there other comparative sets of data like this? We don't know if it's me or you who's frozen.
Yeah, so the primate aging database that I mentioned has a bunch of species, including macaques. So, and 𝗂 does do annual knockdowns, and they're probably like similar to the sanctuaries in that they're intermediate, they're provisioned, but they live like in a naturalistic setting. But chimpanzees, I would argue, are even better because they live longer and they're physiologically more similar to humans. So I think that this is a real benefit, is that we have access to the blood data from the sanctuaries. I'll mention that there are a couple of other sanctuaries that do similar work. So there's a couple of sanctuaries in Central Africa with chimpanzees and gorillas. And so there's actually a big network within the sanctuaries. And because heart disease is so common in captive apes, it's a big goal of this network to understand, even if they don't get atherosclerosis, how does that relate to other types of, like, can you predict fibrotic heart disease, for example? Hopefully we're still stable internet.
Yeah, I caught your answer on that. Yeah.
Great.
Was there any other questions? I think we've just got 2 minutes left. I think we're done. Great talk, Megan. Thanks so much for spending your time sharing that with us.